Hi, my name is Ava Cinealis. I'm a senior at the University of Wisconsin-Stout, and I'm studying Applied Biochemistry and Molecular Biology. These past few semesters, I've been studying protein structures of prion disease, and recently, the protein that causes chronic wasting disease in deer. The same protein that causes chronic wasting disease in deer can also cause mad cow disease and scrapie in sheep. People can inherit a prion form that causes Creutzfeldt-Jakob disease in humans. We are concerned that these proteins can infect humans. Mad cow disease was actually the cause of about 200 human deaths from eating contaminated beef. Living in Wisconsin, chronic wasting disease is something that we should all be concerned for. In fact, Wisconsin is a hotspot for chronic wasting disease, as there have been 43 known counties with infected deer. So with this being said, if you're hunting in Wisconsin, it's probably smart to get your deer tested, as scientists are not yet sure if they can infect humans. Not that long ago, it was thought that cells were required for an organism to be infectious, like bacteria and viruses. However, prions have been discovered to be the first infectious protein. Recently, a protein related to Alzheimer's was caused to found disease in patients who were treated for an endocrine problem. We are interested in being able to predict how likely a prion protein is to be infective. For a prion to be infective, it has to misfold. We are developing semi-quantitative measures to predict its tendency to misfold. Generative AI is changing the speed of research. The software AlphaFold uses artificial intelligence to predict the protein structure from its amino acid sequence. We have found that AlphaFold is picking up on subtle changes in protein sequence within a specific motif of the prion protein that varies slightly across a handful of mammalian species. The structural predictions AlphaFold is making is generating protein secondary structure that is strongly reminiscent of the structure of misfolding prion proteins. Some of these species we are studying are more vulnerable to developing an actual prion disease, like the cat and Syrian hamster, while other species are thought to be less sensitive, such as the dog. We can break the predicted structures down into whether they have alpha helix or beta sheet content. The healthy protein will have an alpha helix and the disease protein will have a beta sheet. In our work using AlphaFold to detect the sensitivity of a given modeled peptide to beta sheet formation, the percentage of beta sheet predicted was measured and plotted against a general overall measure of susceptibility of the species estimated from the cited literature, and we did find a correlation. We are currently exploring the potential predictive power of these trends, especially for prion protein forms where we may not have structural information. There is still so much research that needs to be done, and I want to thank you for being interested in protein structures of prion disease.